All right, so it's 9 o'clock. Welcome everyone. Thanks for joining us at the weekend. And then welcome to our today's Immune Zoom seminar. So it's absolutely my pleasure to welcome our today's speaker, Dr. James Chou from Shanghai. So James actually currently is a principal investigator at Interdisciplinary Research Center of Biology and Chemistry, Shanghai Institute of Organic Chemistry, Chinese Academy of Science. He is also currently served as deputy director of the IRCBC at the National Key Laboratory of Chemical Biology of Shanghai Institute of Organic Chemistry. So I think James actually did really wonderful work during his career, and then he did really a lot of breaking through discovery. So a very, very brief introduction about James. James actually received his undergraduate degree in physics from University of Michigan, and then he continued to pursue his PhD training in biophysics in Harvard. And then afterwards, he continued to get training as a postdoc in NIH. Actually, we sort of at the sort of like a colleague in Dr. Adrian Beck's group in NIDDK in NIH. So from 2003, James actually moved back to Harvard to start his independent research program, and then all the way promoted to tenured professor at 2012. So after about 10 years, in 2023, he decided to again switch his career path and then move back to China and then join Shanghai Institute of Organic Chemistry. So James' group actually has been engaged in the research of the signal transduction mechanism of transmembrane receptors. I think mainly cytokine receptor and the fusion mechanism of virus and the host membrane. So his lab actually has developed a series of biochemical and biophysical technologies to review the blind spots of these the proteins in the transmembrane and near membrane regions, and discovered a lot of important new biological functions and concept. The research goal of his lab is to understand the spatial temporal organization of the immune and cytokine receptors on the cell surface, and the mechanism of their regulation of regarding to their downstream signaling pathways. And then his group actually really made a lot of discovery of new immunotherapy regulators and multi-specific drug delivery systems. Because his remarkable scientific contributions, James has received numerous awards, including Pure Scholar Award in Biomedical Sciences, Smith Family Investigator Awards, and then Genzyme Outstanding Achievement in Biomedical Science Award. So I think today James is going to tell us a little bit about his work on the biophysical and biochemical work regarding the cytokine receptors. And then I think without further ado, thank you so much, James, for joining us. And then we are very much looking forward to your talk.
Okay. Can you hear me well?
Yes. Yes.
Okay. Thank you, Tran, for the invitation, for introducing me to this Immune Zoom interest group. Especially also thank you to those in North America for staying up so late to hear my talk. So I as Tran mentioned, I wasn't really trained as an immunologist, but have collaborated with many immunologists, great immunologists. I was trained in biophysics and have long been interested in the problem of transmembrane signaling. So for the past several years, my lab has been exploring the functional roles of transmembrane domains of cytokine receptors, which are normally the inconspicuous regions of receptor biology. And I hope my talk today will help explain some of the aberrant signaling from immune or cytokine receptors. So let me first begin by showing the widely accepted model of cytokine receptor activation. So this is the ligand-dependent activation in which ligand binding to the active domain causes receptor oligomerization, which then triggers high-order assembly of the downstream signaling complex to fully activate the signaling. Although we still don't have any structures of the transmembrane, entire transmembrane signal zone, the principle of high-order clustering is more or less established from various imaging data. The role of the transmembrane domain in this model, however, is often not clear other than being simple membrane anchor. But there have been several, actually very early studies suggesting that the TMD, or transmembrane domain of single-pass transmembrane receptors, can serve an active role in receptor activation. And here I just show a few examples. This early paper from Bob Weinberg's lab at MIT was probably the first to report the importance of TMD in receptor signaling. They found that a single valine to glutamine mutation in the TM of HER2 resulted in ligand-independent receptor activation, as shown here in the lower panel here. I'm also showing on the top the activation model of ligand-induced dimerization of HER1, which is EGFR, also known as EGFR, that is essential for signaling. Now, this paper was really striking at the time because the ligand of HER2 was unknown and still unknown today, actually. So exactly how this single mutation in the TM can drive receptor activation remains elusive. Here's another example, somewhat different, from Harvey Lodish's lab, also at MIT. They discovered for the erythropoietin receptor that a single mutation losing to serine in the TM allowed viral envelope protein to drive receptor oligomerization and ligand-independent activation. Know that the viral protein in this case is itself a TM dimer, and it can only activate the mutant EPO receptor, but not the wild-type receptor. Another interesting example is the thrombopoietin receptor, TPO receptor, whose activation is also strongly dependent on the TMD. In this case, the Engelman lab at Yale showed that the FDA-approved drug, L-Trombobag, can actually target the receptor TMD and could serve as some type of molecular glue that induces receptor dimerization and activation. So in all these cases, detailed structure information are still lacking. So one of our goals was to dig deeper into the membrane regions of this receptor to see what else have escaped our attention. So I also want to mention how I became interested in immunology. So about 20 years ago, I got into a collaboration with Kai Wuprofenik at Dana-Farber to study the structural basis of TCR-CD3 assembly, which, as you all know, largely mediated by interaction among the transmembrane domains. And that was really the first time we explored the use of NMR and the bicells to look at membrane regions of single-pass membrane proteins. So together with Chengqi Xu, who is at the Shanghai Institute of Biochemistry and Cell Biology, and also Matt Call, who is currently at Weihai, Australia. So we made a couple of fascinating findings. For example, the cytoplasmic item motif of CD3 epsilon is buried in the membrane, and so it is actually sequestered by the membrane from phosphorylation. So as some sort of a safety mechanism. And also with MedCall, we discovered actually how charges within the TM transmembrane region, which is normally considered unstable, can actually drive transmembrane domain assembly and stabilize them. So encouraged by these studies, I thought there was a need to further develop biophysical methods to fill the structural gaps in the membrane of immune and cytokine receptors. Okay, so a little bit on the methodology for studying TM transmembrane domain oligomerization. So we all know that structure determination of the intact single pass TM receptors is very difficult because the different domains across the membrane are not rigidly connected. So it is difficult to do single particle averaging with cryo-EM, for example. So here are some of the frequently used methods for studying the TMDs. And the one, of course, you all know is the biochemical method involving typically, alanine or cysteine scanning mutagenesis, and then reading out oligomerization using, for example, assays such as the bacteria to hybrid assay, or you can also use other type of FRET or split GFP type of assay too. So this is a common method that biologists like to use. In the middle is a solution NMR study of TMD in deterging micelle, and this can also be quite effective, which is exemplified by the work of Engelman and co-workers on the glycophorin A transmembrane dimer. This was an important study because they discovered the glycine XXX glycine motif for the transmembrane helix dimerization. And this is a well-known motif in membrane biology. So, of course, deterging micelles are sometimes not compatible with TM helix oligomerization, so caution needs to be taken. Lipidic cubic phase crystallography is another option, although getting well-diffracting crystals can be a lot of work. And my lab has been developing the use of medium-sized bicelles for NMR studies of TM interactions. So bicelle is a self-assembled lipid disk comprising long-chain lipid and short-chain detergent. Its size can be tuned by the lipid to detergent ratio, which is also known as the Q ratio. Thus, according to this formula, for example, at larger Q, the bicelle is bigger. So previous NMR and the small angle X-ray scattering data here indicate that at Q around 0.5 is the transition point between bicelle being mixed micelle-like and lipid bilayer-like, suggesting that NMR samples should be made with Q around 0.5. And fortunately, bicelles with Q around 0.5 can normally give high-quality NMR spectrum, as shown here for the transmembrane trimerization of the Fas receptor, and also for the transmembrane heterodimerization between the IL-2 gamma chain and IL-7 here, or the IL-9 receptors. Here you can see it's very it's interesting, nice to point out that the red peaks correspond to are from the gamma chain, the blue peaks are from the 7R or 9R receptors. You can see the chemical shifts are actually for the red peaks are somewhat different, suggesting they're paired with different the private members of the family. So thus we took advantage of the bicelle system and developed a rather streamlined protocol for determining oligomeric TMD structures. The protocol involves 4 steps shown here. first reconstitute TMD in medium-sized bicell, and the step 2 is biochemical methods for determining oligomeric state of the TMD in bicell. Then the step 3 is NMR experiments for measuring intermolecular distances and also computational methods for searching for assembly of the TMD complex that agree with the experimental data. Then finally, in step 4, the membrane partition of the TMD is determined by a method called the paramagnetic probe titration analysis, which I'll show a little bit later. So using this method, we aimed at performing I would say, a comprehensive survey of TMD structures from the TNF receptor superfamily. And the reason for this is that one, TMD structures of this family was essentially unknown. And two, there are also very puzzling mutations associated with ligand-independent receptor signaling that are either in the TM or in the membrane proximal region. Most of you have seen this mechanism. We all know the mechanism of the general canonical mechanism of TNF receptors. TNF receptors are mostly activated by trimeric ligand. That is, binding of the trimeric ligand can trimerize the receptors, and then the trimeric complex further somehow can aggregate into larger signaling complex. Now, our goal was to investigate whether the TMDs have any intrinsic property to oligomerize in this mechanism. So thus far, we have looked at the TM structures of 5 members of the TNF receptor superfamily. And interestingly, they don't all assemble to trimers as we initially thought, which would be consistent with a trimeric ligand. For example, the FAS receptor and the TNFR1 here, they form trimers. The 41BB, for example, TM is monomeric actually. The TM is not oligomeric. That was unexpected, but also unexpected for DR5 and TNFR2. The transmembrane domain actually forms multimers of trimer. So today, I'll focus on the DR5 because it's TMD alone can form higher order clusters. So does that mean the TMD can actually drive receptor activation? Right, so that was a question, was very puzzling at the time. So we went ahead and tested a number of hypotheses. First, we look at the structure. The DR5 TM clustering is actually structurally specific. So this is not just the hydrophobic, non-specific hydrophobic sticking or interaction within the membrane. It is actually very specific. This slide shows that on the right here, the trimeric assembly is actually mediated by the threonine XX alanine sequence motif, whereas the dimeric interaction is mediated by the GXXXG motif, which I just mentioned. So we found that single mutations in these motifs could completely disrupt the trimer or dimer interaction. So we then tested the functional implication of these mutations that disrupted TMD oligomerization. We found using 2 independent cell assays that, in this case, using stable cell and the transient transfection cells, we found that mutations, single mutations that either break dimerization or trimerization completely abolished ligand-dependent signaling. So that was really amazing. This result indicate, and also you can see here, we even tested a no effect mutation. So this valine 227Y did not really influence TMD oligomerization, you can see it behaves just like a wild type. But any single mutations that breaks dimer or trimeric interface completely abolished signaling. So this indicates that TMD clustering is actually an essential part of ligand-induced receptor signaling. But does this also mean that TMD clustering alone could drive signaling if not physically constrained by the ectodomain? So then we did another experiment to test this hypothesis. We designed experiment in which we inserted a TEV protease site between the ectodomain and the TMD, and examined if the receptor can be activated by proteolytic removal of the ectodomain by the enzyme. So the answer is yes. We showed that first. We showed on the left both cleavable and wild type DR5 could be activated by trail, which is the ligand, but only the cleavable DR5 could be activated by the enzyme in the absence of trail. And this activation correlated well with the shedding of the ectodomain from the cell surface. So the result indicates that in the absence of ligand, the receptor ectodomain could adopt some sort of autoinhibitory state or conformation. Exactly what this conformation is, I'll come back to this later. But before that, I'll just do a quick summary for the first part. Is that we show that the bi-cell NMR method is actually generally applicable to structural studies of transmembrane domains. And importantly, using this method, we found that the TMD of DR5, at least, if unconstrained by the ectodomain here, it can actually form specific high-order clusters in the membrane and directly drive downstream signaling. So, and this is probably likely true for some other family members whose TMD can also form high-order clusters. Now, for the second part, I'll continue with the TMD and come back later to the receptor pre-ligand association. Second part, so we were wondering, so is TNF the only family that shows this type of high-order clustering, or shows really the active role of transmembrane domain in signaling? So we then asked what other cytokine receptors or what receptors from other cytokine receptor families may have this sort of property. And the one family we became interested in is the common gamma chain receptor family. And this family is interesting because each of the members, upon ligand binding, must specifically pair with the common gamma chain to allow signal transduction. It is known that gamma chain sharing is mediated by the ligand binding. The question is, do the transmembrane domains also contribute to receptor sharing at the membrane level? So to address this question, Cai Tiantian and Rachel Capello conducted a comprehensive survey of TMD dimerization using the bacteria 2 hybrid assay shown on the left. I won't have time to describe the details. This is a well-established assay published by a group in France. They found, so Tiantian and Rachel found that the TMDs in the gamma chain family show extensive heterodimerization. And the red checks here indicate the heterotypic TMD interactions we have tested thus far. So as you can see, they match remarkably well with receptor sharing network. So as a negative control, we even tested the gamma chain TMD interaction with the interleukin-5 receptor TMD from the beta chain family, and they don't interact. So this suggests that TMD interaction is actually family specific. So this was quite remarkable and puzzling, considering that the gamma chain transmembrane helix is really just a simple TM helix. How is it possible for a simple helix to have the sophistication of recognizing receptor TMDs with highly diverse sequences? So that was a question behind the structure study. We then performed full-scale NMR structure determination of the gamma chain TMD in complex with the IL-7 receptor and IL-9 receptor transmembrane domain. So comparison of the two structures revealed a knob into hole mechanism. So this is quite classic mechanism in structural biology. This mechanism for TM helix sharing, that is the gamma chain transmembrane helix has an isoleucine knob here that fits into a hydrophobic pocket of 7R or 9R. We have shown that such mechanism is applicable to all other two-component receptor sharing, including the 4R and the 21R. But the three-component assembly of IL-2 receptor is somewhat different. So we then tested the implication of knob into hole in the signaling, in receptor signaling, whether this is essential or not. So as you can see here, mutating the gamma chain knob, which is isoleucine, which is a mutation isoleucine 273 to tyrosine, almost completely abolish step 5 phosphorylation. Likewise, breaking the 7R pocket with the mutation leucine 255 to tyrosine also abolished signaling. And as a negative control, we also mutated residues on the opposite sides of the interface, and they did not really influence the signaling. So this knob into hole packing is really critical for at least in the case of IL-7 and IL-9 we have tested so far. They're critical for ligand-dependent signaling. So what can we do with this knowledge? Then we did something I thought was pretty cool. So using this new structural knowledge, we explored whether it is possible to address some of the disease mutations in the TMDs from this family. And the one such TM mutation reported for the IL-7 receptor is the valine 253 to glycine mutation. So this is a gain of function mutation that causes ligand-independent 7R activation and the T-cell leukemia. So by solving the NMR structure of the mutant TMD, we found that this V2G mutation led to TMD hormone dimerization on the opposite side of the gamma chain interaction interface. So, and this explains why 7R can actually oligomerize without gamma chain and ligand. So we then asked the question, can we then design a transmembrane helix to reverse this aberrant signaling caused by the TM mutation? So as such, we used the TMD, we just borrowed TMD from the 7R and introduced this gain of function mutation to block 7R hormone dimerization in this case, right? So because this peptide does not have the signaling component, so it won't signal, but it'll block the 7R hormone dimerization. But at the same time, we introduced the leucine 225 to Y mutation on the opposite side to disrupt the pocket that interacts with the gamma chain TMD so that it does not interfere with ligand-dependent activation. And remarkably, it actually worked. As you can see here on the left, this is in the in the absence of ligand, you can see that the mutant V253G indeed shows very substantial tonic or ligand independent signaling. This was observed in the absence or presence of gamma chain. But then adding the design peptide completely suppressed the tonic signaling. And on the right here, we also show that adding the design TMD did not really affect ligand-dependent signaling. So this was really, we were also quite surprised that the design of design peptide can really fix or repair, if you will, restore the normal control of the IL-7 receptor signaling. So that was quite cool, I thought. So to summarize the second part, we found that receptor TMD interaction can be highly specific, even allowing, in some cases, the design of TM helix to fine-tune signaling. And for future studies, we will look further into other gain of function as well as loss of function SCID mutations in the TM of gamma chain. For example, some of the gamma chain loss of function or SCID mutations within the TM actually cannot be well explained by our current heterodimer structure, suggesting that because we haven't solved the structure of the IL-2 complex yet, so suggesting that the TM complex of IL-2 beta and the gamma might be different. And so for the third part, I now go back to DR5 and discuss how can the pre-ligand state of DR5 be autoinhibitory. What type of structure features that is responsible for this autoinhibitory mechanism? And this study was conducted by Du Gang, Zhao Linling, and Anissa Belfeltmi. So one of the technical problems for this type of study is that the isolated DR5 ectodomain in solution is monomeric. So in order to reconstitute the system more accurately, so we anchored the ectodomain to liposome blended with nickel NTA lipids using the his tag as affinity tag. So the idea is to anchoring the ectodomain onto the surface of liposome pays for the rotational entropy cost. So actually it will promote the clustering on the membrane. Then the N-terminus of this ectodomain was labeled with Psi3 or Psi5 for FRET readout. Indeed, we found upon anchoring to the liposome, strong FRET was observed, indicating self-association. Then after confirming that the ectodomain can self-associate in the absence of ligand, we suspected that such pre-ligand association may be inhibitory. So we tested 5 nanobodies to see if disrupting the pre-ligand association with nanobody can actually activate receptor. So all 5 nanobodies shown here have very high binding affinity for DR5, but only nanobody 1 and 5 caused significant drop in the FRET, indicating at least partial disruption of the DR5 cluster. Now, in the absence of trail, nanobody 1 and 5 could only induce very modest activity, about 20% at high concentration. We then explored possible synergy between different nanobodies for further dissociating pre-ligand cluster, and found that some of the combos here indeed could further disrupt signaling and induce stronger activation. And on the right here basically shows a summary of all the data. This is combining all the data. We can see obvious positive correlation between disruption of pre-ligand cluster and the receptor activation. So this suggests that at least at the high expression level, DR5 exists on the cell surface as some sort of the pre-ligand cluster that serves a autoinhibitory role. So dissociating in this case, the autoinhibitory cluster can lead to receptor activation. So this got us really curious about the structure of the pre-ligand association. We then used similar lipid anchoring approach by anchoring the ectodomain, DR5 ectodomain to bicelles. You can see that in the absence of bicell here on the left, the ectodomain gave a really beautiful NMR spectrum in solution, consistent with being monomeric. Then upon mixing with bicelles, a subset of peaks gradually moved over time until the protein began to precipitate after about 7 days. And then we show by this cross-linking experiment that the time-dependent peak movements were really due to continuous clustering of the protein on the bicelles, shown here. So then by performing full-scale NMR structure analysis, we found 2 different interaction interfaces, two different modes of interaction responsible for pre-ligand clustering. And this is also, it is also interesting to mention that some of these residues shown here in the 2 interaction interface happen to correspond to the peaks that have shifted most. And this suggests that the 2 modes of interactions were weak and dynamic in the beginning, but it became more stable over time as the cluster grew bigger. So since the 2 interaction interfaces are on the opposite sides of the DR5 ectodomain, higher order clusters can be envisaged. Now, if we just simply replicate these 2 interactions, the larger clusters could look like multiple ring-like structures. But of course, this is purely hypothetical at this point. The only way to really verify this is by either super resolution light microscopy or just getting a cryo-EM structure for this cluster. Another important aspect of receptor structure is how they are positioned on the membrane. So we addressed this question by performing this experiment called a paramagnetic probe titration experiment, which I mentioned earlier. In this case, the lipophilic paramagnetic probe used was 5-DSA, which is just a fatty acid with nitroxide spin label at the C5 position. Then titrating 5-DSA to bicell anchored DR5 ectodomain generated dose-dependent decay of NMR signal. Now the middle panel here shows 3 examples representing different proximity to the bicell, and they're mapped onto the structure on the right. So the data shows that in contrast to our common depiction of receptor standing up on the membrane, the DR5 ectodomain actually rests flat on the lipid bilayer in a specific posture with its ligand binding or trail binding site partially buried. So this was also unexpected, if you wish. So this, of course, so far all the structure studies have been done on a pretty artificial system of this, NMR system with bicelles. Another question is the NMR structure also represented on the cell membrane? So at least to partially address this question, we implemented a split GFP assay using a more native construct, including the ectodomain, the stalk sequence, as well as the monomeric form of the TMD. The reason to have the TMD being monomeric is that we do not want to introduce other confounding variables that could influence ectodomain clustering, because TMD itself can also cluster. Here, as you can see, the wild type protein show very strong puncta of split GFP signals, whereas introduction of mutations that disrupt either interface 1 or 2 significantly reduced GFP by 4 or 5 folds. We also tested combination of here, the bottom, combination of nanobody 1 to break interface 1 and the mutations to break interface 2, and this led to the most dramatic dissociation of the cluster. So this data suggests that NMR-derived structures of the pre-ligand association are also most likely also represented on the cell surface. And this is a final data slide showing just to understand whether, how did the nanobody actually disrupt the pre-ligand association. We just performed a very quick, epitope mapping, antibody epitope mapping by NMR chemical shift perturbation. We found that the nanobody 1 binding site overlap here very strongly with interface 1 of the pre-ligand association, but now with the trail binding site shown on the right. So the data suggests that a compound, so this compound could be small molecule if you wish, in principle can weaken pre-ligand compound that can weaken pre-ligand association should sensitize the receptor to its endogenous ligand if it does not block ligand binding. Okay, so this to summarize the third part. So at least for DR5, we found that receptor in pre-ligand state does not stand up as often depicted in the literature. Instead, it lies flat on the cell membrane and forms clusters to prevent ligand-independent signaling driven by the TMD or intracellular domains. I think this auto-regulatory mechanism is particularly important when the receptor expression level is high. So to conclude, I just want to do an overall summary. The first is the unexpected role of receptor TMD in driving higher order receptor clustering and activation. Now, receptor TMD dimerization is actually nothing new. We have seen this in many other cases, even before our study, EGFR, TRK-A, or TPO receptor. But the function of TMD in driving large-scale receptor clustering and activation was really unexpected. And it could be a more general phenomena worth further exploring. Second is, although we have determined many TMD oligomer structures, I was still struck by the structural specificity of TMD oligomerization. How a simple transmembrane helix can afford so many different features for the diverse and the specific helix recognition within the membrane. It is so specific that we could even design TM helix to specifically influence receptor signaling. So this is also something that could be further explored. Now, finally, receptor pre-ligand association may serve a general auto-inhibitory function, especially when they are overexpressed on the cell surface. We believe this property could be further explored for developing more specific receptor agonist or antagonist. Okay, I want to acknowledge the following people for the work I have presented today. Most of this work was done at the Harvard Medical School. So these people were from my lab at the Harvard Medical School. On the left column are including Matthew Cobb, Bill Fu, Jody Dev, Wayne Chang, Alessandro Pia, and Chen. They actually contribute a lot for developing the biophysical methods for studying TMDs. Then people on the right column, Leslie, Tian, Rachel, Ling, and Gan, Anissa, those were people, postdoc who actually did the work which I presented today. And as Chen mentioned recently, last year I moved my lab back to China, to Shanghai Institute of Organic Chemistry. So some of the peptide TM design work, which for restoring the interleukin-7 receptor signaling, was done by Ming Chen and Du-Ling Yu. And we also collaborated with Hao Wu, Richard Siegel, as well as the Sanofi immunology team. And then I also want to thank the funding, both from the US and China, and also the NMR facility from MIT Harvard Center for Magnetic Resonance, as well as the Protein Science Center here in Shanghai. So thank you for your attention. I'll be happy to take any questions.